Okay, well, thanks a lot for the opportunity. I'm always very happy to talk about my leeches and what are we doing with them. And first of all, I'm Alejandro Manzano Marin. I'm of Mexican origin, but I'm working here in Vienna since some years ago, and I work in the Department of Microbial Ecology. Although I'm actually much more of an evolutionary biologist interested in symbiosis interactions, how symbiosis interactions really shape biology and evolution of their hosts. And this is how I got eventually to leeches, even though most of my career I've worked on insects. Then now I'm working on leeches, and hopefully through these three organisms that I'll present in the next 5.5 minutes, you get a nice idea of why we're working on these leeches, what is the utility of their genomes, what are we going to use them for, and what kind of questions do we want to answer. So the first genome that I decided to present was Helobdella europaea. So all of these have some sort of connected goal, but also kind of its individual goals. And Helobdella europaea, it's this nice leech that you see here to the right. I put just a map of the world because this is actually an invasive species which has a very wide distribution that we're still yet to uncover. So every year or so, there's a couple of more papers that show that there's actually species is now being recorded from a new place in the world. So probably very worldwide widespread. So who is this leech? So this leech is member of the Glossifoniformes, which are dorsoventrally flattened leeches, which hold a proboscis instead of a jaw, like most leeches that many of you might be familiar with from ponds in Europe. It's also, as I told, it's an invasive leech, which probably came originally from South America, from an unspecified source or sources, and then it has being spread out throughout the world through the aquarium trade. It is a predatory leech, so it's not that the leeches you might be very used to, which are the leeches that feed on blood, but rather it feeds on invertebrates. And this is where you can see why the aquarium trade has served a lot to spread around the world, since it usually comes in tanks because it has been infecting snails or other invertebrates. So what is very interesting about this leech is that it belongs to then an order where we know it has actually lost the ability to feed on blood. So it's one of the representatives that have repeatedly lost the ability to feed on blood. And it also shows, like all members of Glossifoniformes, parental care, which is a unique trait evolved in this order of leeches, which means that they're going to deposit their cocoons on their belly. These cocoons are going to clothe into leeches, and then they're taken to the first feed. What are we using its genome for? Where we're using it along with further ongoing genome sequences of different predatory leeches to explore, let's say, divergent systems where this ability to feed on blood and digesting blood has been lost, to kind of see what, let's say, makes a non-blood feeder or what makes a blood feeder within this group of animals. It's also hopefully going to contribute to the understanding of different biological features such as parental care or the body shape. And this is also going to serve to be through comparative genomics with other non-invasive closely related Helobdella species to try to see which kind of biological traits have actually allowed it to spread all over the world. We know that one of them is probably that they're able to reproduce asexually, so you can start whole populations with one individual. So we're going to start exploring this through obviously different kind of experimental systems, which we have it actually living in the lab. The second leech that I'm going to be talking about is Placobdella costata. So this is a Palearctic species, up to recently the only one that was known of the Palearctic region of this particular genus, which is mostly widespread in North America. It looks like this little guy right here. Why is it interesting? Well, it's also a member of these Glossifoniformes leeches, so also dorsoventrally flattened. As I said, until recently was the only nominal species known from the Palearctic region. Very recently was described as a species complex, which we're also working a little bit with throughout a large network of people interested in leeches across Europe. It also displays, contrary to the last leech that I showed you, displays bacterium, so a completely novel organ that has evolved to house symbiotic bacteria. So bacteria that in this case are going to be providing B vitamins are most certainly vertically inherited and show this reductive genome evolution. So this you can see that why it's very interesting also to contrast it to the previous species. Also, similar to the other Glossifoniformes, displays parental care. So this is something that we're also going to try to look at between blood and non-blood feeding species. What is an impact and future directions of this? So currently and through the start of sequencing from this species, we actually now built a nice large network where we're going to do further sequencing of the different species within the species complex to also analyze genomically the biodiversity or the diversity that we have of this species here in the Palearctic region. It's going to also contribute to the understanding of development of biological features such as parental care, body shape, but also develop like the novel evolution of organs such as the bacterium, along with microscopy and a lot of micro CT studies, and also according to potential host microbe interactions and metabolites that might be shared with its simian to try to control this production of B vitamins after feeding. Lastly, I'm going to talk to you about the one that a lot of you might be more familiar with, which is Hirudo verbana. So Hirudo verbana shows a distribution mostly in the southern eastern part of Europe, which is this blue area right here. It is not a dorso-ventrally flattened leech, but a tubular one belonging to the Hirudiniforms, or where a lot of the world's medicinal leeches locate. It has also, for my interest, which is symbiosis, it shows a very low complexity stratified bacterial microbiota, which is very interesting to try to find and understand digestive relationships with bacteria, because it's not very complex, but it's also not very simple microbiota. We know that its current taxon might some of its species level diversity, such as for the last species, is something that we're also now exploring through ongoing projects, and it's easy to read in laboratory conditions. And just to wrap it up, this is also part of further ongoing sequencing with a lot of other groups that we're going to try to also analyze antimicrobial peptides and its functions across the Hirudo genus through ongoing further genome sequencing. We will also use this genome to explore different populations of these species and to understand the diversity of their microbiota through nanopore adaptive sampling using the reference chromosome level genomes. I also want to further, hopefully, the understanding of leech's microbiota in general and its role in digesting and maintaining a blood-based diet. And that's it on my side. Thanks.